Hello, welcome to this teaching video. My name is Therese Bergfors, and I run the crystallization platform at the Department of Cell and Molecular Biology here at Uppsala University. Today I'm going to give a lecture on how to crystallize proteins. And to understand why this topic is of importance and it being included in a course on structural biology, let's take a look at some statistics from the Protein Data Bank. Most of you have been using or are at least familiar with the Protein Data Bank, which is a repository for structural information. It's called Protein Data Bank, but of course it includes all biological macromolecules. So it includes viruses, nucleic acids, ribosomes. But let's just call it the Protein Data Bank. Now, as at the time of the recording of this video, there are 168,000 structures deposited in the PDB. To put this in perspective, let me mention that in 1984, when I started my career at the crystallization platform, there were exactly 100 structures in the PDB. So this gives you an idea of how progress has been made in this field over the last 40 years. And of all these structures that have been deposited, the overwhelming majority, 89% of them, have been determined by one particular method, and that is X-ray crystallography. It accounts for 89% of these structures. Now, there are other methods that are also used for structural determination, and these include nuclear magnetic resonance, NMR, and they also include an up-and-coming method, cryo-electron microscopy, cryo-EM. The first structure solved by cryo-EM was deposited in 1991, and in 2017, this method was awarded a Nobel Prize. But by far and away, the majority of the structures are still done by X-ray crystallography. So there are three vocabulary words or three definitions that I need to make clear here at the start of the lecture so that you can follow what I'm going to say. The first of these is precipitant. And it's real unfortunate that precipitant and precipitate are so similar to each other in English, because the precipitant is a chemical entity that's used to drive the protein out of solution. The precipitate is the protein, or a solid phase of the protein. It can be ordered or it can be disordered. We want the ordered precipitate, which is known as a crystal. So if you can just remember the difference between precipitant and precipitate during the course of this lecture, you'll be doing fine. We have to define what is a crystal then. What do we mean by a crystal before we go into how we do it, how we get proteins to crystallize? A crystal is a repeating 3D array of molecules. So shown here is a crystal lattice containing protein molecules. And crystallization is the act of getting liquid, it's a phase transition of liquid phase protein to the solid phase as an ordered 3D repeating crystal lattice. Now, if you're going to do a structural determination by X-ray crystallography, here are the steps that are involved. The first one is to get purified pure protein. How much do we need? Well, 5 milligrams would be a great starting point, but even 1 milligram will sometimes work. So let's say between 1 and 5 milligrams of protein. Pure protein, it needs to be about 90% pure. The next step after you get the pure protein is to put it into crystallization screening. More on that later. We do large screening trials to find the right conditions to give crystals. Once we have crystals, Then we can put them in the X-ray beam and collect data. Now, these steps of data collection, data evaluation, model building, refining the 3D structure, these are no longer rate-limiting steps. The real bottleneck is still the crystallization, for reasons I'll go into. But let me give you an example of how things have changed since the beginning of my career. The first protein I worked on was P2 myelin, and it took 2 years to get that structure. It took maybe 150 crystals. It was a 24/7 job, meaning once you put the crystal in the X-ray beam, you collected day and night, you came in on the weekends, you changed the film, you developed the film by hand. So it took a long time. Nowadays, by comparison, a data collection that took weeks back in 1984 takes less than 1 minute at a synchrotron beamline. In fact, it's terrible. You don't even have time to go out and have a cup of coffee before you have to come back and change the crystal and start the next data collection. So these steps of the data collection and so on are no longer rate limiting. They go very fast. The problem is still getting the protein to crystallize. Now in this slide, I'm showing the same information in the previous one, but just in a different way. I'm showing it here as a bar graph. If you start with, say, 100, 120 targets that you want to crystallize and solve the structure, you begin by cloning them, and most of them can be expressed. You have a slight number that aren't soluble. You lose a few here. These aren't soluble. And then you lose a few more because you can't quite get enough material. Can't purify enough. You can't purify that 1 milligram. So you lose some there. But here, as you can see, the big drop-off is between this step and this one, getting the good crystals. In fact, only about 20% of the proteins that you start with are soluble and will crystallize. There's a big drop-off right there. But here's the good news. This is the good news, this bar there. If you manage to get good crystals, you can almost always solve the structure nowadays. Those mathematical problems or limitations in interpreting electron density, those have been solved. So it would be, in fact, an extraordinary situation that you had a good electron density map and you couldn't solve the structure. But what do we mean by good crystals? Well, by this we mean the crystal has to diffract to at least 3 angstrom. If you want to see ligand binding, it needs to be even better resolution, about at least 2.5 angstrom. And the same protein can crystallize in many different polymorphs, many different morphologies. So if we look at number 1 here, what's wrong with these crystals? In fact, these are stacks of crystals piled on top of each other, and we can't really use them in the X-ray beam. It has to be a single crystal in the X-ray beam, and it would be very difficult to separate these. So that's why in picture 2 here, this actually represents an improvement. At least now, these are single crystals. We have to fish these out of the droplets, so it needs to be a single crystal that goes into the X-ray beam. Now, if we change the conditions, we can change the morphology. These crystals, in fact, diffracted much better than these, but they still didn't diffract well enough. In fact, the ones that we did use in solving this particular structure were these crystals in number 4. And these diffracted much better because they were bound to a ligand, a fatty acid. Ligands will often help stabilize the protein and improve the diffraction. The thing to remember, there's 2 things actually to remember from this slide, and it's not the size of the crystals. It just so happens that these were bigger than these. Just because it's bigger doesn't mean it necessarily diffracts better. These are maybe better looking, but the appearances don't have anything to do with it either. You can have really ugly looking crystals that diffract great, and you can have very beautiful crystals that don't diffract at all. The appearance of the crystal is not what's important. The only thing that really counts in defining a good crystal is that how well it diffracts. All right, so I've mentioned that only about 20% of these proteins that you start with actually will produce crystals. So why are proteins so hard to crystallize? There's many reasons, but today I'm going to discuss what I think are the two major ones. It's the nature of the search problem. You can't predict the crystallization conditions for any protein initially. It's because it's a multi-parametric search problem in which there are many solubility parameters that have to be examined. And not just in isolation from each other. They're interactive effects. So if you change the pH and the temperature, you will also change the solubility differently than if you just change the pH or you just change the temperature. In fact, there's at least 30 different solubility parameters that I could have put on this slide. And maybe 28 of them don't make any difference whatsoever for this particular protein. Your job, your search for the needle in the haystack is to find let's say, the two solubility parameters that are important. So to do these multi-parametric searches, we have to do large crystallization screening trials. Many experiments are required, and in this particular picture, I'm showing our old crystallization room in which we appropriated mail sorting boxes from the building and stacked them against the wall here from the floor to the ceiling. And inside each one of these mail slots, there is a tray, one or two, maybe three trays. And inside each tray, there is anywhere from 96 to 384 experiments, screening experiments. Let me give you a close-up of one of the trays. So this particular tray contains 96 conditions or screening solutions, and has two slots times 96 for putting the protein. Nowadays we have automation to help us with this screening problem. Here I'm showing our crystallization hotel. This is where we barcode the plates and stack them into this hotel, which has an automated robot arm to put them on a regular schedule into the imager, which you see there behind me. Here's the imager. This imager takes a series of pictures. It screens each plate 13 times over a period of 2 months. After that, if nothing's crystallized, then it has to leave the hotel and make room for more promising experiments. Now here you see the lab manager. She doesn't look so happy in this picture. I think she's having a software issue that day. But let's take a look at what these images that the imager takes, what they might look like. So for this example, I got this from my friend Joe Luft, who is the lab in charge of the crystallization platform at a high throughput institute called Hauptmann Woodward, which is located in Buffalo, New York. A high throughput facility might do easily 900 of these plates per month. At our academic platform, we do about 900 plates per year. So they have a lot of examples, and the one I'm going to show you is to illustrate how just very small changes in the pH give quite different outcomes in terms of crystallization. Everything in this series is held absolutely constant. The protein concentration, the precipitant concentration, the only thing we're changing is the pH, ever so slightly. So at the pH 4.5, yes, the protein has come out of solution, but it's come out as a rather disordered precipitate. If we tweak the pH a bit, this actually shows an improvement. If you look here, it may not look like an improvement, but in fact it is, because if you look here along the perimeter, there are very small crystals actually forming. Now when we change the pH just a bit more, we can see that now we are getting crystals and much less of the precipitate. Here at last, we can actually get some crystals that we could put into the X-ray beam. For instance, this one, and maybe this one. They have slightly different morphologies. We should test both. Now at pH 5, the crystals are getting smaller, and at 5.1, they're gone. So you can see just a small change from pH 4.5 to 5.1 can have quite different consequences. This brings us now to the second big reason why it's hard to crystallize proteins, and that is the nature of proteins themselves. The proteins, once they're purified, have to be stable over the course of the experiment. These crystallization screening trials take anywhere from a day up to 2 months, sometimes more, before the crystals, if they're going to appear, do appear. If the protein is not stable over this time course, then you aren't going to get it to crystallize. So the protein, there are big demands on the protein to be stable. Many proteins are not, they break down. Another problem with the proteins is that they often have multiple conformations. They have loops, they have flexible domains. Maybe they have the ligand bound, maybe they don't have the ligand bound, and when they bind the ligand, they change shape. They're very dynamic molecules. And I'd like to show an example of one such case, which is this protein, it's a cellobiohydrolase, a cellulase. These are proteins that hydrolyze cellulose. Hydrolyze these cellulose change. Now this protein we spent at least 10 years trying to crystallize. Not just our lab, other labs too. Now that we know the structure, it's pretty obvious why it was so hard to crystallize. But at the time we didn't. What makes this protein so hard to crystallize is that it's extremely flexible. For the large hydrolytic domain is 465 amino acids, but it's attached to a very flexible long rope or tether of 30 amino acids, which in its turn is connected to a clamp of 30 amino acids. Now let's say if you look at me, and this is the big domain, the hydrolytic domain, and my arm is the rope, and my hand is the clamp. You can well imagine that a protein that is doing this 24/7, looking for some cellulose to clamp onto, is not going to pack easily into a crystal lattice. There's simply too much movement in it. So how to deal with this problem? Well, this was done back in the dark ages, before molecular biology was even invented. So we chopped the protein up using proteolytic enzymes and then tried to crystallize the pieces by themselves. I call this the divide and conquer method. Nowadays, we would simply clone different domains of the protein and try to crystallize them separately. Then you put the whole picture together. Now, I really like this example because of this particular cellobiohydrolase, because we had to use, in fact, 3 different structural methods to get the final picture. So for solving the big hydrolytic domain of 465 amino acids, this one, we used X-ray crystallography. We were able to get crystals once we cut it off here. Now this piece, the rope, was solved by small angle X-ray scattering, SAX. This isn't a good method for globular proteins, but it works very well for long fibrous proteins. For instance, like silk. Yeah. And then this small clamp of 30 amino acids was solved by nuclear magnetic resonance. Very small proteins are also difficult to crystallize, but they work fine for NMR. So I like this example because it's a hybrid. It uses 3 different structural methods to get the overall picture. The methods don't compete with each other. It's not use SAX or X-ray. They complement each other. They each have applications that are good for certain problems. Okay, but let's say you're lucky, you've gotten crystals and in your screening, and now your problems are still not over. You have to get that crystal out of the droplet and into the X-ray beam. So to do this, we use something called loops, and we say we go fishing with the loops. Here is the loop. These crystals are grown in very small drops. This drop is only 0.2 microliters. It's sealed. So to get the crystal out, we're going to have to take the seal off of the experiment and go fishing with this loop. And as you can imagine, a drop that's only 0.2 microliters is going to dry out very fast indeed. So you have to have great skill and be fast. That crystal has to get out of the drop and into the liquid nitrogen quickly. Now, why are we using liquid nitrogen? This is to prevent a problem at the synchrotron. Radiation damage. There's 2 kinds of radiation damage, of course. The primary damage, which the X-ray beam knocks off the electrons directly from the atoms, there's nothing we can really do about that. It's a law of physics. However, once you knock the electrons off, this generates free radicals, which then propagate throughout the entire crystal. They knock off more electrons, and now you have those electrons knocking off more electrons. This is the secondary damage. This we can't prevent either, but what we can do is slow it down by working at cryo temperatures. So this is why we put the crystals into liquid nitrogen and keep them at minus 210, minus 196 degrees centigrade. So we are collecting these we're collecting the diffraction patterns of crystals that are under liquid nitrogen. Okay, once you get your crystals into the loops and into the liquid nitrogen, we pack them into shipping dewars and send them off to a synchrotron, where we use the X-ray beam. And this one, in this particular picture, I'm showing you the synchrotron at Max 4, which is located in Lund, Sweden. That ends part 1 of this video tutorial, and it's time for a coffee break. Why don't you take 30 minutes or so, get yourself a cup of coffee, and you can watch a film during that period. It's a 4-minute film. Ideally, what we would like to do is take you on a tour of our crystallization platform and show you how we set up the drops manually, which is what some of you will be doing in the lab exercise this afternoon, setting them up manually. And the video also shows how we do it with the crystallization robot. So take 4 minutes and some coffee and watch that. Then welcome back for part 2 of the video in about, let's say, 30 minutes. Thank you.